Hello. Good evening. Thank you, everyone, for coming. My name is Eric Lindstrom, and I'm a senior in political science here. I have a strong interest in the philosophy of science, and so last semester I helped Dr. Hector Avalos and the Committee on Lectures plan tonight's event. The topic of intelligent design has been prominent in our schools and universities across our country. This is evidenced by the statement made by President Bush last August and by the Dover trial in Pennsylvania last fall. Our campus has seen much discussion of this topic as well. The Iowa State Daily had about 40 articles and opinions last semester on intelligent design. And I stopped and checked my Facebook before coming, and the Flying Spaghetti Monster has just under 400 friends tonight. I'm currently taking an honors course taught on the issue, taught by three professors, Dr. Hector Avalos, Jim Colbert, and Michael Clough. Last fall, these three professors co-wrote a statement on intelligent design that was eventually endorsed by some 400 faculty members of Iowa's three regent universities. It became perhaps the largest national faculty statement on intelligent design. All agreed that educating faculty, students, and the public on the unscientific nature of intelligent design was of utmost importance. So tonight we bring you a long-awaited lecture on why intelligent design is not science. First, though, I'd like to thank our co-sponsors. I'd like to thank the Lectures Committee, which is funded by GSB, the Department of Biochemistry, Biophysics, and Molecular Biology, the Department of Chemistry, the Department of Geological and Atmospheric Sciences, the Department of Mathematics, the Department of Philosophy and Religious Studies, the Molecular, Cellular, and Developmental Biology Interdepartmental Graduate Program, and the Atheist and Agnostic Society. And I've also been told that after the event, our main speaker will be doing book signing for a bit. But now I will let Dr. Hector Avalos of the Department of Philosophy and Religious Studies introduce our main speaker, Dr. Robert Hazen.
Thank you, Eric. For months we have been pondering, I and some of my colleagues, how best to educate the community about intelligent design. And we have considered a number of formats, including a forum with both sides. Usually the other side does not want to participate. We've considered a panel discussion. And in September, I encountered an editorial by Dr. Robert Hazen in the Chronicle of Higher Education. So I sent him an email along with two of his colleagues, and he graciously accepted the invitation to come. So I am very pleased to have him here. Dr. Robert Hazen is the Clarence Robinson Professor of Earth Science at George Mason University. He is a scientist at the Carnegie Institution of Washington's Geophysical Laboratory. He received his Master of Science in Geology and his PhD in Earth Science from Harvard. He is the author of 300 articles and 16 books, including Why Aren't Black Holes Black? Also Genesis, the Scientific Quest for Life's Origins, and a number of other very notable works, including a lecture series on the joy of science. So please join me in welcoming Dr. Robert Hazen.
Thank you, Eric, and thank you, Hector, very much, and thank you all for coming this evening. This is such a pleasure and an honor for me to talk to you about this subject, which I find so fascinating and which I think we all agree is very much in the news today. Genesis. That word evokes so many passionate responses about the processes that led to life's origin. For many people, the first chapter of the Bible provides the most compelling origin narrative. Others evoke a purely natural origin of life. And there's certainly no subject that instills greater tension between science and religion than the origin and evolution of life on Earth. The prevailing scientific approach to life's genesis, and the theme of my new book, is called chemical evolution. According to this hypothesis, life arose by a totally natural process of emergent complexity. And I must tell you up front that I'm persuaded that life's origin was completely consistent with natural law. This hypothesis, like any good scientific model, leads to numerous testable predictions. Life must have emerged as a sequence of chemical steps of increasing complexity. Each of these steps, as we shall see, must be testable in the laboratory. Many hundreds of researchers now devote their lives to this experimental pursuit. But as you all know, intelligent design represents a rival hypothesis. According to supporters of this model, life is much too intricate, too complex to have arisen by any natural chemical process. Life requires hundreds or thousands of interconnected molecular reactions. Take away any one of those critical molecules and life fails, so it's difficult to imagine a stepwise process from simplicity to complexity. Indeed, according to this view of life, life is irreducibly complex. The logical conclusion, the ID proponents say, is that some supernatural designer, either an alien or God, must have engineered life on Earth. In this view, life arose by some combination of natural and supernatural events. Now, this invocation of supernatural processes makes one wonder whether intelligent design is a valid scientific approach. On the one hand, ID does obey one requirement of science, in that it makes predictions. It predicts, for example, that no natural process could have produced the eye. It predicts that no natural process could have produced the bacterial flagellum. That's the little squiggly thing that helps bacteria move. It predicts that no natural process could have produced human consciousness. And of course, no natural process could have produced that first living cell. Scientists can, at least in principle, test these predictions. If we can go into the laboratory and show a stepwise process by which simplicity becomes the complexity of an eye, or the complexity of a bacterial flagellum, or indeed the complexity of the first living cell, then we have disproved, we have invalidated one of the predictions of intelligent design. On the other hand, intelligent design fails a key science litmus test because it relies on supernatural processes. This is another example of the old God in the gaps argument. If some natural phenomenon is seemingly too complex to understand, so the story goes, then we must resort to the supernatural. That's not a very satisfying explanation, and more to the point, it's not a testable explanation. As a result, most scientists and the federal courts have concluded that intelligent design is not science. And that leads us to the so-called debate. Those individuals who are philosophically opposed to the seemingly random, undirected scientific explanation of the evolution of life on Earth take great comfort in intelligent design. We are special. And it's more special to be the creation of a caring designer than the product of random events. Thus, many people, including the president, advocate teaching the debate between evolution and ID. It's only fair, they say, to present both sides of the story, as if this was some evenly balanced pair of choices. At the same time, virtually every scientific leader in society of science objects. For example, the American Chemical Society says intelligent design should not be taught in high school biology classes as an alternative to evolution. And I can understand, at least superficially, why such objections lead many non-scientists to the conclusion that scientists are closed-minded, that we're unwilling to consider alternative points of view. So what are scientists to do? How do we respond to intelligent design? Ultimately, the most powerful response to any God in the gaps argument is to fill in the gaps. So, we want to design a research program that demonstrates a natural transition from chemical simplicity to emergent complexity. If we can do that, if biological complexity can be shown to arise spontaneously as the result of completely lawful, natural processes, then we have effectively disproved the central tenet of intelligent design. The origin of the prehistoric monument called Stonehenge provides a good analogy. Some people have claimed that Stonehenge is much too big and too complex to have been constructed by Stone Age peoples, so they invoke space aliens as the builders of this monument. They say the alignment is much too perfect, the astronomical alignment, so you couldn't possibly have done it in ancient times. But look, if we can demonstrate a straightforward process, however difficult and time-consuming, by which a primitive culture could have made Stonehenge, then there's absolutely no reason to resort to space aliens. And you can do the calculation. It would take roughly 1,000 able-bodied people working for many years. So many, many thousands of man-hours, maybe millions of man-hours, but it still is possible. Look, we can never prove that space aliens didn't help to build Stonehenge. Archaeologists can't do that. But it's logical to accept the simpler, more familiar explanation. For origin of life researchers, the simpler explanation rests on the amazing field of emergent complexity. Complexity often emerges from the interactions of numerous individual particles such as molecules, sand grains, cells, stars. The collective system displays behaviors not associated with individual particles. And in my book Genesis, I argue that the origin of life is the quintessential emergent phenomena. Indeed, life's origin can be modeled as a sequence of emergent chemical steps, each of which adds a degree of organization and complexity. So let's take a closer look at some emergent systems. One of my favorite is slime mold. Slime mold lives most of its life as a single-cell organism. Sort of looks like an irregular amoeba-shaped thing. Under certain situations, though, temperature and humidity, these cells send out chemical signals. And they create gradients of chemicals so that other cells are attracted and they start clumping together into some mass of cells. And then they actually begin crawling across the forest floor. And ultimately, these form stalk-like structures. And at the tops of the stalks, new spores are released and a new generation of slime mold occurs. Now, no one of these individual cells knows that it's part of this collective phenomena. These individual cells are just reacting to local chemical signals, and yet the emergent phenomenon is extraordinary. Another example you all know about is ant colonies. Individual ants send out signals, chemical signals, perhaps a half dozen or more, pheromones. These signals indicate the presence of food, the presence of danger, other sorts of symbols. And so ants, they tend their young. They gather food. They do amazing things. They build colonies of incredible three-dimensional complexity. They tend aphids, like shepherds. They do other collective behaviors that are truly extraordinary. And no individual ant knows that it's doing this as a collective whole. It's just responding to local chemical signals. And then the most amazing emergent phenomena of all, human consciousness. Your brain holds trillions of neurons. They're connected to each other with various neural networks, and electrical impulses pass through. And so you have this incredible neural network, incredibly complex in its intricacy and organization. And what emerges is consciousness. You're all conscious this evening and paying attention to this, but no individual neuron knows that it's doing this. It's just part of the collective. The emergent phenomenon is consciousness. And I would argue that life is the quintessential emergent phenomenon. We see these phenomena all around us all the time in space. We see them in the rings of planets. We see them in the typical formation of an exploding star. We see them in the spiral arms of galaxies. On Earth, we see it in the behavior of molecules forming crystals. We see it in the level of sand grains forming dunes and ripples. We see it in larger structures such as hurricanes. We see it at every scale of life. The scale of molecules forming cell membranes. We see it at the scale of individual cells forming slime mold, for example, and of course of individual organisms forming collective behaviors. And even in human society, agriculture is a beautiful collective example. The various athletic competitions. And I love the example of a symphony orchestra. Many individuals, each doing their own thing, collectively making music that no one person could make. These are emergent phenomena. They occur all around us all the time. It is a consequence of our natural world, and life is the quintessential example. So here are my central assumptions. Origin of life research. Now remember, what we're trying to do is create a continuum, a stepwise progression from simplicity to complexity. If we can do that, then we have basically disproved the prediction of intelligent design that life is too complex to have arisen by a natural process. I argue that the first life form must have been chemical in nature and was based on organic molecules, just like today. I argue that the origin of life used the most simple of raw materials, the oceans, the atmospheres, rocks, and minerals. And I argue that the origin of life required a sequence of emergent steps, each one increasing the complexity of the whole system. Now, I say Genesis occurred simply in 4 steps, and this is the organization of the book. The first, you had the emergence of biomolecules. Second, the emergence of organized systems of molecules. The third step is the emergence of sets of molecules that make copies of themselves, self-replication or reproduction. And finally, natural selection kicks in. And most origin of life scientists would argue that the transition from non-living to a living world occurs when you get competitive systems of molecules that begin to evolve by natural selection, a much more rapid process. So let's look at these. Genesis 1, the emergence of biomolecules. The idea that you make simple organic molecules from raw materials, the volcanic gases like carbon dioxide, carbon monoxide, water. They come together and they form molecules that have carbon-carbon bonds, 2 carbon atoms, 3 carbon atoms, larger and larger molecules, the basic building blocks of life. And these experiments have been done for more than 50 years. First experiments, 1953, University of Chicago. Stanley Miller, the young graduate student. Harold Urey, the Nobel Prize-winning mentor. Together they designed this apparatus, which is nothing more than simple water, a gas mixture, and electric sparks that simulate lightning. So you have the ocean, the atmosphere, and lightning going on in this glassware. And lo and behold, after just a few days, that solution turns colors and oozing black gunk off those electrodes. And you find the molecules of life emerge. Amino acids, the building blocks of protein. Sugars, the building blocks of carbohydrates. You find fatty acids and other fats and lipids that make cell membranes. You find nucleic acid bases. Those are building blocks of DNA and RNA. Amazing. For all the world, this looks like a nice, well-balanced diet. And so scientists were amazed. They were awed, and they said, this must be the way it happens. But it's not the only way it happens. There are many other ways to make molecules. For example, in deep space, in what are called dense molecular clouds, there's a little chunk of the Eagle Nebula, which is called a dense molecular cloud, but by dense, it's much greater vacuum than anything we could produce on Earth, maybe 1 million molecules per cubic centimeter. Ultra cold, too. So we go into a laboratory, we design a chamber, which is vacuum and extremely cold. You take molecules, you irradiate them. Here's Lou Amendola, who leads the NASA Ames Research Center in California, where this group is working. There's his apparatus. And as you irradiate these ices, as you irradiate these molecules, gradually larger and larger structures are built. You make amino acids, you make sugars, you make alcohols, you make the molecules of life in deep space. Deep space is littered with the molecules of life. And then my own interesting hypothesis having to do with the deep sea hydrothermal vents. Here's another environment where there's chemistry and there's energy coming together to make molecules. The idea that deep on the ocean floor, where volcanic fluids, hot volcanic fluids come out at high pressure, also deep underground, perhaps even here below the state of Iowa, we have chemical processes going on at high temperature and high pressure. Now, this idea of the hydrothermal systems is not without its controversies. Stanley Miller, in particular, doesn't like it. He says the proposal is based on misunderstandings because the high temperatures of the vents would destroy rather than manufacture amino acids and other molecules. Well, we say the high temperatures of the vents create the synthesis, and then those molecules are exuded into the cooler ocean, and they survive and go on to contribute to life. Stanley Miller in 1992 was more blunt. He said the vent hypothesis is a real loser. I don't understand why we even have to discuss it. But of course, as a scientist, we do have to discuss it. We have to do experiments, and that's exactly what we do at the Geophysical Laboratory. We do hydrothermal organic synthesis, taking our chemicals, welding them into small gold tubes, taking those tubes, subjecting them to crushing pressures, high temperatures. My colleague in much of this work was Hat Yoder, who designed the laboratory for high temperature and high pressure, And even at the age of 75, long after he retired, he was still in the lab every day, still working, still part of our team. When he died a couple years ago, it was a great loss to our work, but his efforts are certainly being continued. And here's the kind of thing we did. Oh, by the way, George Cody, my colleague at Carnegie also, George Cody is an organic geochemist, and he does the analysis of these molecules. He tells us what we've made. So, here's one process that we know goes on in hydrothermal vents. You start with a very simple molecule like natural gas or methane, carbon with 4 hydrogens, and you build longer and longer chains, step by step, just by adding CH2 groups, and you make longer molecules. And that's the game here. We're trying to make larger molecules from small molecules. There's also another process. This is called Fischer-Tropsch synthesis, by the way, but there's another process that goes by the fancy name of hydroformylation. Just means instead of inserting CH2 to make your chain longer, you insert CO. And that's another way that you make longer and longer molecules. Now, in our experiments, when we tried to do Fischer-Tropsch synthesis, boy, this really goes by gangbusters. You put carbon dioxide, you put hydrogen and water in these gold capsules. You put a little bit of iron metal in as a catalyst. Simple conditions, 300 degrees centigrade, about 500 times the atmospheric pressure, and for 24 hours. And lo and behold, we get an explosion of molecules, long chain-like molecules, 20 or 30 carbon atoms long, starting just with carbon dioxide. This is an abundant process. Here's this other process where you insert carbon and oxygen to make your chain longer. And to make a long story short, thousands of experimental runs later, using all different kinds of minerals, sulfides of cobalt and nickel, iron and copper, lead and zinc, we find that both of these processes, making longer chains by inserting CH2 or by CO, occurs. And so you make a diversity of molecules, a wealth of different kinds of molecules. Add a CH2 here, add a CO there. Make a branch, make a ring. All different kinds of building blocks that may have relevance to the origin of life. An embarrassment of riches. And what we can conclude is that the synthesis of these molecules in a prebiotic environment was relatively easy. And as a card-carrying mineralogist, I'm delighted to say that minerals obviously played a key role in this origin. This part is easy. This part we know. Genesis 1 is pretty well understood. But the next step is much more complicated, and that's taking these molecules, selecting just the right ones, making larger molecules called macromolecules. See, the problem is that the prebiotic synthesis is so easy that you make huge numbers of different kinds of molecules. But life is idiosyncratic. It only uses a few kinds of molecules. Most of them it rejects. Most of them are useless to life. So what process might have led to the selection and the concentration and the organization of those molecules? We know that in some cases that this occurs naturally through self-selection. In other cases, we may need minerals. Let me give you some examples of the idiosyncrasies I'm talking about. Amino acids. These are amazing. Now, in your bodies, the proteins that you eat, they have 20 main amino acids, different molecules that are all closely related to each other. But if you look in meteorites, if you look from space, there's more than 70 amino acids that come down, and of course, chemists know of hundreds more. And these are very specific kinds of amino acids, not just any amino acid will do. And what's more, on top of that, amino acids come in left-handed and right-handed versions, mirror image versions, but life only uses the left-handed ones. It throws away the right-handed ones. How did this selectivity come to be? Here's another example. I showed you the Fischer-Tropsch process. This is how we make, in industry, long-chain molecules. Add one carbon molecule after another, so you have 1, 2, 3, 4, and so forth. But life uses a completely different technique, something called the fatty acid synthase. It adds carbon atoms 2 at a time. So you go 2, 4, 6, 8, and you end up with chains with even numbers of carbon atoms. That's a diagnostic signature of life. If you ever have a whole bunch of long chain molecules, if there's 2, 4, 6, 8, the even numbers but not the odd numbers, it's very likely something derived from life. This is the kind of idiosyncrasy that life shows us. Well, so how do we get selectivity in this situation? One of the ways turns out to be self-organization. Let me show you an experiment that really yielded some surprises for us. I took a very simple molecule, 3 carbon atom molecule called pyruvate. I put it in gold capsules with CO2, which has 1 carbon, and the thought we had was 1 plus 3 is going to equal 4, and we'll make some nice 4 carbon molecules out of this Conditions very simple, 200 degrees centigrade, 2,000 atmospheres, just 2 hours. Well, boy, were we surprised. I expected just a little bit of colorless liquid. We got a puffed up capsule, and you open up, bang! It pops, it releases a huge amount of internal gas pressure. And then shortly after that, you get this foaming and hissing, and this yellow, oily brown goo starts crawling out of the capsule and coating the bottom of the vial. And then you start smelling this stuff. And at lower temperature, like 200 degrees, it sort of smells like molasses. But as you get up to around 300 degrees, it smells unmistakably like Jack Daniels. And I tell you, people come by the laboratory and say, "Where's the party? Can I have some?" It's remarkable. You're making some really interesting molecules here. This isn't just a four-carbon atom thing. These are interesting molecules. So something's happening, and you do an analysis, and you see an explosion of new molecular species, hundreds, perhaps thousands of new molecules forming from just the simple reaction of pyruvic acid. So what's going on here? How in the world could this help us in the origin of life? We're making so many different kinds of molecules. They can't possibly be of any use. And yet it turns out there is a use. If you take this yellow oil goo and you put it in water, it does something quite amazing, something called self-organization. This work has been done with Dave Deemer, my colleague at Santa Cruz, also Marilyn Fogel at the Geophysical Laboratory. By the way, you notice that all these scientists are smiling. Origin of life research is really fun. We're all smiling a lot in this. And here's what happens. You have certain kinds of molecules that have one end that is attracted to water, but the other end is repelled by water, a molecule called an amphiphile. And when you put lots of these molecules in water together, they find a way to nestle up to each other so that the water-loving ends are on the outside and on the inside. The water-hating parts are as far away from water as possible, and you form a membrane, a bimolecular membrane with two layers coming together. This is called a vesicle. And lo and behold, you put those molecules, that yellow-brown oily goo, into water, and it forms these exquisitely beautiful fluorescent green vesicles. They range from about 10 microns up to 50 microns. They have an inside and outside. They look very much like a kind of primitive membrane structure. This happens spontaneously, and we know this would have happened on the early Earth. Unless you think this is some kind of magic, that this doesn't happen very much, this is actually something we're all pretty familiar with, because making this kind of circular structure we've been doing since childhood. It's really quite delightful. I love the opportunity to play with this toy. But also you see self-organization happening, molecules. Also you see how fleeting self-organization can be, and isn't that true of life as well? One of the aspects of emergent phenomena. Okay. Not all molecules self-organize. Many molecules, amino acids for example, dissolve in water and they stay dissolved in water. You need some other technique to pull them out. So I've been thinking about this specific problem of how did those left and right-handed amino acids get selected so you concentrated left-handed and got rid of the right-handed ones. Now the motivation for this is because many of life's molecules have this characteristic of chirality. There's left-handed and then there's a mirror image right-handed molecule. And of course we want to know how life became homochiral. But there's another motivation. I've been working with chemical engineers at Carnegie Mellon because this is a $200 billion, that's billion with a B, dollar industry of making chirally pure pharmaceuticals. If you take Zocor or Lipitor or Xanax or any one of a whole wealth of drugs that many of you or your friends take, a lot of the money you're paying for those has to do with this chiral purity. They have to be handed pure, and there's a good reason. See, chiral purity is important. Here's a simple molecule called limonene. The right-handed version smells like oranges. The left-handed version smells like lemons. It reacts differently because our bodies are chiral. There's another example. This just came out of Nature a couple of weeks ago, and I think this is amazing. Frontalin. It's a molecule that's secreted by male elephants as a sex attractant. Now, the reason chirality is important is that young males apparently produce more of the plus form than the minus form. They're not as attractive as the older males that produce a 50-50 mixture. That 50-50 mixture repels other males and attracts the females. And if that isn't important, I don't know what is. And finally, a rather more sinister example is the thalidomide story. The right-handed form of thalidomide cured morning sickness, and so was taken by many pregnant women several decades ago. But tragically, the left-handed form causes birth defects. And before they had chirally pure thalidomide, this drug therefore caused this unintended consequence because chirality is important. So here's the question. Prebiotic synthesis produces equal mixtures of left and right-handed molecules. Life demonstrates this incredible selectivity. What was the process by which left and right was selected? And my hypothesis is that perhaps minerals played a role. Minerals play a role because they have chiral surfaces. The most common chiral mineral is quartz. It comes in left and right-handed forms. The structures have helices that either go to the left or to the right. So you have two-handed forms. The question is maybe quartz selects left and right-handed molecules. And all previous workers took these beautiful quartz crystals and ground them up to a fine powder. It increases the surface area. It lets you do more of an intense experiment. The trouble with that is that it really destroys the possibility of learning what's going on at a detailed scale. Here's a specimen of quartz that I collected when I was 19 years old, Paterson, New Jersey. And if you look carefully, you'll notice that there's a red coating, a very pale red coating on some of these crystals. And indeed, some of the coatings occur on some faces, but not on others. In other words, the adsorption of materials is face-dependent. Each face has a different kind of adsorption. If you grind up your crystal, you destroy the possibility. So what I decided to use big crystals that have nice big faces so we can study the chiral absorption. Feldspar, one of the commonest minerals in nature. Pyroxene, for example, diopside, another extremely common mineral, the commonest iron-bearing mineral. And calcite, by far my favorite. Calcite is a crystal that occurs in snail shells and clam shells. It's extremely common in biology. It also has abundant chiral faces. And so we did experiments on calcite. I did these experiments with my dear friend, Glenn Goodfriend. Glenn Goodfriend was a master of analyzing amino acids. His most famous projects were with Stephen Jay Gould, and tragically both of these dear friends died within a few months of each other 3 years ago. A great loss to science. But what I did with Glenn is take a crystal just like this. I had a beaker with 50% left-handed amino acid, 50% right-handed amino acid. Just simply dumped the crystal and let it sit there overnight. Came back the next day, pulled the crystal out, rinsed it off, washed it off, and then carefully, face by face, put a little bit of acid that strips off the amino acids. Take that acid, put it in a vial. And you can analyze, therefore, the left and the right-handed molecules as they come right off the crystal. And lo and behold, what we found was that left-handed amino acids stick more strongly to the left-handed faces, right-handed amino acids stick more strongly to the right-handed faces. Minerals, in fact, can select left and right-handed molecules. Now, this is a very tedious job to take each crystal and each molecule and stick it in and do these experiments. So we were looking for a faster way, and so look at our smiling friends, Jake Mall and Andrew Steele, molecular biologists working with DNA fingerprinting techniques. What they use is a technique called chip writing. This is used to analyze, for example, genetic diseases of large numbers of different sequences of DNA. But we use it for a different way. This is a machine that puts tiny dots of liquid on flat surfaces. So we take a mineral surface. We decorate it with hundreds or thousands of tiny dots of different liquids. And you can do up to 126 mineral chips simultaneously in the same experiment. You then look, by rinsing off, you see what sticks to which surface and what doesn't. This is a great way to study thousands of mineral molecule combinations. The traditional way of analyzing these is called chip readers that looks for fluorescent markers. The spots that are fluorescently tagged show up brightly in fluorescent light, and you can do that. Here is a fluorescently tagged amino acid called lysine on left-handed quartz on the left, right-handed quartz on the right, and you see that this sticks much more strongly to the right-handed quartz, a suggestion of a chiral effect. But there's a problem here because fluorescent tags themselves are big molecules, and they didn't occur in the prebiotic Earth. We're only interested in the lysine, not in that fluorescent tag. So we have to find another technique, and we find it in something called time of flight. secondary ion mass spectrometry, or TOF-SIMS. Working with my friends at the Smithsonian Institution, we are now able to analyze whether an amino acid sticks or doesn't stick to a surface, or we can look at sugars, we can look at other molecules, and this opens up a whole new world of studying not just one molecule and mineral at a time, but thousands of pairs. Progress is certainly being made, and what we're finding is indeed there's incredibly important selection of molecules by minerals. So my conclusions of the second chapter of Genesis is that prebiotic molecules can indeed be selected. They can be concentrated, and it occurs both by self-organization and by absorption on mineral surfaces. But you all know that's not a step in the life. And remember, we're trying to examine this hypothesis that we can show a stepwise process from simplicity to complexity, from the geochemical world to the biochemical world. And so we have to go beyond just making molecules or organizing those molecules. We have to have self-replicating molecular systems. Reproduction is a key aspect of life. Now, how did this happen? Some people say it happened with a cycle of molecules, like the familiar citric acid cycle that's part of every cell in our bodies, also known as the TCA cycle. And what this is about a dozen molecules that forms a cycle. Let's start with the 6-carbon atom citric acid. You split it into two atoms plus 4 atoms of carbon. And each of those molecules starts another cycle. So you end up with 2 citric acids, then 4, then 8. It's a self-replicating cycle. Also, the branching molecules off the side give us new amino acids, they give us lipids, they give us sugars, they give us other molecules of biological interest. So this is an engine of biochemical synthesis. This is a kind of holy grail. If we could make a citric acid cycle work in the laboratory under primitive conditions, that would be a great discovery. Now, not everybody sees it exactly this way. Stuart Kauffman is another person who has been thinking about this. Oh, I should show you Harold Morowitz, my great colleague at George Mason University. He's the person who has most articulately advocated this idea of the citric acid cycle. But Stuart Kauffman, a network diagram, he talks about autocatalytic networks of molecules. And this is a large collection, maybe thousands of molecules, each one of which helps to synthesize, helps to catalyze a different molecule in the network. So each molecule contributes, and you have a closed group, a closed network, which gets richer and richer and richer at the expense of all that other useless molecules on the side. It becomes a self-replicating network of molecules, which then jump-starts more and more efficient cycles. That's another idea that's out there. Now, we've been trying to do kind of combining these, looking at citric acid and seeing what network of molecules might occur. We get very complicated diagrams. The details are sort of mind-boggling, but we actually are very close to having a closed system of molecules right now, and this is fruitful research that we're continuing. So there may be a network of molecules that's related to the citric acid cycle. But I have to confess, there are many people who think that this is totally the wrong track in origin of life research, and that the first self-replicating molecule is not a cycle, but in fact is DNA or RNA-like. It's a genetic molecule. This idea has been supported, especially in the last 20 years, since it was found that RNA, simple genetic molecule, not only can carry information and pass it from one generation to the next, but it also can catalyze, it can promote reactions. So an RNA molecule could do it all. It could be the genetic molecule and the metabolic molecule at the same time. So we know that all cells use RNA. RNA is an extremely ancient molecule, and this has led to what's called the RNA world hypothesis. But there's a problem. It turns out that RNA is implausible as a molecule. There's just no way in a prebiotic environment to make it. It's just too delicate. It's too complex. It involves fitting together too many parts. So there's no way to synthesize it. And this leads to the question, what happened before the RNA world? It turns out in the origin of life field, it's perfectly okay to come up with wild ideas, to speculate. As long as you have good chemical intuition, you can kind of be a little fanciful and tell stories and then say, what about this? Let's try it out. Let's experiment. I want to tell you about that. Here's Dave Deemer again with my good friend and colleague Nick Platts. Nick Platts was a graduate student. He was 40 years old getting his doctorate degree at RPI. And in the book, I tell the story about how Nick was just about to be deported. He only had a month left to finish his PhD thesis, and he hadn't started after 5 years. But then he had this incredible idea. And the idea is so steeped in real chemical intuition ideas that people have really embraced him. Nick got his doctorate, and he's now working as a postdoc trying to establish these results. Here's Nick Platt's PA World, and this gives you an idea of the kind of creativity and the fun that we have in the field. This has not been proven, but it's still an interesting idea. PAHs, polycyclic aromatic hydrocarbons. They're ring-shaped molecules composed with 6 carbon atoms in a ring, and these interconnected rings form all sorts of structures that you can see the diversity of these things. Hundreds and hundreds of different kinds of PAHs. And they are abundant in the universe. If you look at the universe in deep space at an infrared wavelength, you will see the characteristic signal of PAHs. That's the kind of glowing in this infrared wavelength that indicates that there's vast quantities of these ring-like molecules. So any planet, any moon is going to have abundant PAHs to work with. So what Nick says is PAHs, it turns out, are just like those membrane-forming molecules. The flat surfaces of the PAHs repel water, but the edges are attracted to water. So if you put lots of these ring-like molecules in water together, they will spontaneously self-organize in a stack, like a stack of pancakes. That was his prediction. Next, he said, the edges are very reactive, so they will accept and concentrate other small flat molecules. Notably, the bases, that is the letters of DNA, A, T, G, and C, those will stack up. Furthermore, the spacing between those bases will be exactly 3.4 angstroms, which is exactly the spacing you see in DNA. Nick's chemical intuition went beyond this. He said it would be very simple under some environments to get a backbone linking together those bases. A backbone, for example, of formaldehyde. And then, under changes of temperature or pH or other conditions, perhaps that whole system would break loose. It would fold on itself. It would actually be able to make copies of itself under the right circumstances. And here you have a self-replicating genetic molecule, something that's a precursor to RNA, the PA world. There's no experimental verification of this. It's, some people would say, wild speculation. But it got him a PhD, and it's a pretty neat idea, and he is testing it now. So this is the kind of exuberance of origin of life research. Lots of really creative ideas. So the conclusion of Genesis 3. We haven't synthesized a plausible prebiotic molecule or cycle of molecules that can replicate itself yet. But I think we're getting very close. It would not surprise me at all if in the next 5 to 10 years, someone at some laboratory would have a system in a test tube that could keep replicating itself. It wouldn't be alive, it would be an amazing chemical feat. And that brings us to the last step, Genesis 4, the emergence of natural selection. Here I have to make the argument. If you have a cycle of molecules that makes copies of itself, organic molecules are so diverse and so intricate that you can have slight variations, slight mutations, if you will, and the cycle will still work. And as you have competing cycles of molecules, they will compete for the limited resources of energy and atoms. And so that competition will drive selection. You'll get ever more efficient cycles. You'll have the emergence of natural selection. Now, I can't tell you all the details. I've got a chapter on this, which is it's amazing work. It's done at Harvard University. Jack Szostak is talking about molecular evolution. He's actually shown how this can work. He puts molecules in a beaker, and through a series of cycles, he actually causes molecules to evolve, to be selected for certain key tasks. This is actually a patented technique now. It's being used all the time for drug design because this is such a powerful technique to evolve molecules when you put a competitive environment on. So this is not science fiction. This is science fact now. My conclusions. The origin of life on Earth is best understood in terms of the sequence of emergent chemical events, each of them adding a degree of structure and complexity to the prebiotic world. We don't know all the details yet. There are vast gaps in what we know. But there's no compelling evidence to suggest that life's origin was other than a natural process. I understand that this scenario troubles many people. How can one believe in a benevolent God if life first emerged by some undirected sequence of chemical reactions? How can we human beings be created in God's image if evolution is an unscripted process of mindless competition? Intelligent design is one answer. We were planned. We were designed. And who did the designing? I know that many proponents of ID refuse to say it was God, but come on, what's the alternative? Some super alien creature? And if so, who designed the designer? I refuse to accept that. Let's be honest, if we speak of a designer, then we're all talking about God as the creator of heaven and earth. But I'd like to close with a different way of thinking about life's ancient origins. Our growing understanding of emergent complexity suggests that life is not some random, improbable event. Indeed, we now know that many emergent steps are hardwired into the very fabric of the cosmos. Given hydrogen atoms, stars follow inevitably. Given stars, the entire periodic table of the elements emerges. And from those elements come Earth-like planets. And now, many scientists are convinced that life is equally a cosmic imperative given a habitable world and sufficient time. And who knows? Perhaps, given abundant life, the emergence of consciousness, self-awareness, and reason are also common in the universe. Science cannot prove, nor can it disprove the existence of God. Science can't prove whether or not the universe is imbued with meaning and purpose. But I firmly believe that science remains our best hope of revealing the majesty of a creation that is learning to know itself. Thank you.
Dr. Hazen will now take questions. If you'd like to form a line at the microphone here in the center aisle, and if you would please be respectful and make your questions short.
I think one of the great challenges in science today is that we only know of one universe and only know of one genesis of life. And so if one is to say that there are many universes, there are many life forms out there, that life has arisen many times, this really becomes more of a personal statement of belief than any sort of scientific statement. I've been amazed in the field of origin and evolution of life how many statements they start with facts, and then you gradually ooze into opinion, philosophy, and theory without much support. So it's difficult for me to say definitively one way or another. Certainly our universe was sufficiently well-tuned that we're here talking about it. And yet, is that an imperative? Is that a chance event? Are we unique? Are we one of billions of living worlds? That we don't know yet. It's a great question. It's one that drives us all. And to me, the most exciting discovery we can make in science in my lifetime would be what's known in the business as second genesis. That would be the discovery of an independent biological origin someplace else, perhaps on Mars, perhaps in deep space. If we could find that, if we could show that there's a second living world out there, that would change our view of the universe incredibly. Thank you.
I would like you to comment on a statement made by a science philosopher, Del Ratch, in his book, Science and Its Limits. The statement is, there are no good grounds, in principle, for barring from science recognition of evidences of design, even if the perhaps unknown fact of the matter is that design is supernatural.
Oh, that's absolutely true in the following sense. Science cannot speak to miracles, to the supernatural, to a designer that we cannot see. There's absolutely no way that I could stand up here and say categorically that we were not the result of a design. All I can do as a scientist is use methodological naturalism, the scientific method, doing experiments that are reproducible by anyone in the world who has the same equipment, make observations of the natural world, use theory in order to come to conclusions. And if I can show in that process, or if my colleagues 100 years or 1,000 years from now can show a process, a stepwise process from simplicity to complexity, then I would say that obviates the need to call in a designer. But it could never disprove the role of a designer. I think science has to remain agnostic on that point. And this is why I say intelligent design is not science. It's a fascinating idea. It's worthy of philosophical discussion. It's important to have forums where we look at different points of view. But science cannot say yay or nay to that. Nor is intelligent design, as it's now proposed, scientific, because it relies on the supernatural.
Since you mentioned methodological naturalism, may I ask a follow-up question?
Sure.
However useful that strategy is in science, methodological naturalism, such a strategy does not constitute a norm that is inviolable, no matter what the evidence. In other words, is it guaranteed that we will never uncover a gap in nature on this planet or elsewhere in the cosmos that rigorous scientific assessment will identify as being bridgeable only via agent activity?
You know, that's a wonderful question, and I really thank you and appreciate that. You know, the idea here is we're all focusing on the gaps, and what is science? Science is limited because certainly there are things we don't know. But when I see a gap as a scientist, I say we start with geochemistry and we end up with biochemistry, and there's this huge gap here. I see that gap and I said, wow, what an amazing opportunity. This is going to be so much fun because there's something we don't know, and I'm curious to see if we can fill the gap. But I have a feeling that some people in intelligent design look and say, oh, that's just too difficult. We just can't deal with that. We're just going to have to throw up our hands and say, let's call in an unseen intelligence. That's a very personal statement for me, and I respect the opposite point of view. But to me, the gaps are an opportunity as opposed to a wall that can't be bridged. There will always be gaps, and the more we know, the more we learn we don't know. That's one of the things about science that's fun and also frustrating. And we can never prove that there aren't gaps that can be filled. So I appreciate it.
Your work is beautiful.
Thank you.
First off, I wanted to compliment you on your work. It's fascinating that you came here tonight. What I wanted to ask you was why intelligent design is not science. Most of what you spoke about tonight was already your work. So based on the title of tonight's lecture, I wanted to ask you, hopefully I'm going to get a chance for a few questions after these gentlemen. So the first one's going to be sort of a lob for you here. I'll throw this up. I assume this is not your first ID rodeo. You've probably been in a debate before. If Dr. ID was here, sitting across the aisle from you, what would be two things as examples of irreducible complexity? Maybe the second most common and like the fifth most common. And what would your answers to those be?
Well, certainly, I gave some examples earlier. The eye is often brought up as an example of irreducible complexity, going from a sightless layer of cells to a fully formed eye. And there's some very interesting theoretical work that talks about that, that you can go from increasing optical acuity just by slightly bending a light receptive surface and then gradually increasing its ability to collect light. And we see all these intermediate stages in nature. Of course, another one is the bacterial flagellum. It's the poster child of ID. And indeed, it's a very complex structure that helps to propel microbes through a kind of rotary motion. And one can argue that without all those pieces coming together, you just couldn't have a flagellum. Well, there are many structures in biology in which there are pieces that have come together, but those pieces have an antecedent purpose. They were used in metabolism, they were used in cell wall kinds of transport and other sorts of functions, and you readapt them and realign them, and they have a different function. Sure, nobody can say exactly how the bacterial flagellum arose now, but we also see some amazing parallels between those molecules and other molecules that had more primitive functions. So we just have to study these systems carefully and try to fill in the gaps. Will we fill in every gap in course of time? No, because the number of things we don't know just keeps growing. The more we learn, the more we don't know. But that's the fun of science, and why shut off that process and just say, too complex?
Thank you.
Hi, wonderful lecture. I'm not a fan of intelligent design myself. I'm a hopeful agnostic who hopes that there is something beyond this world. And I think it would be fair to say that theoretically nothing is impossible, and we could conceivably sometime in the future come up with something that could suggest something about God or whatever you call it. But my question is, do you believe that the proponents of intelligent design have jumped the gun, and that this is really not about God or the existence of God, but it's really about Christianity and politics? And how much do you really think that is the case?
Well, I certainly think there are many dimensions to this debate, as you know. The dimension that disturbs me most, the one which I am most passionately trying to fight, is the idea that it's a dichotomy, that either you believe in God or you believe in science, and there's no middle ground. That's very sad. I think many of my friends and colleagues who have professed some leaning towards intelligent design, I think they're struggling with this because they see in intelligent design a possibility that science and faith can come together. I see it slightly differently. I see that in the incredible power of emergent complexity, not irreducible complexity, not just saying it's so complex a designer had to be there, but emergent complexity, a universe that somehow leads inexorably from simplicity to complexity. I see great power and beauty in that. I see a kind of majesty, and if one wishes to believe in a creator, how better to know the creator than to study creation? I think science and faith can come together in this way, as opposed to these false dichotomies that are created so often by the media, by the fundamentalists on both sides. And let's face it, there are scientist fundamentalists as well as religious fundamentalists. They polarize the debate. They make this so much harder than it needs to be. Thanks for the question.
Agreed. Thanks.
I'm kind of like the last guy. I'm an agnostic, which means the idea of a God is only as absurd as the idea that there isn't a God. And towards the end of your speech, you said something like what is this, aliens? No, like you sort of went, come on. But how much of that, given the complexity of the universe, how much of that stems from your religious beliefs and need to believe rather than hard fact?
What I'm referring to there is just the idea of intelligent design sort of walking away saying, we're not being religious here, we're just proposing another scientific explanation that involves a designer. We're not saying who the designer is. I think that really is a little disingenuous. I think it's very hard to talk about intelligent design the way people do without really at bottom believing that it's God. I'm not saying that God exists or doesn't exist. What I'm saying is if you ascribe to intelligent design, then you're ascribing to a belief that God has intervened in the natural world in a miraculous way.
I guess I'm asking, why does God enter before a more supreme intelligence like aliens or something?
Well,
may I ask what your religious affiliation is?
Well, let me put this in two parts.
Okay.
the idea of the alien is perfectly plausible given the intelligent design scenario that maybe there is an alien, but then you're talking about a super supreme being that's much more advanced than we are. It's almost unknowable by us. And so how did that super supreme being come? Was it a natural process by which it evolved, in which case it makes the scientist's job that much harder? Or did God create the alien intelligence, in which case you're back to God? You have to start somewhere. And as far as my own religious beliefs, it's sometimes hard for me to know whether that's truly relevant to a discussion like this. But
I was just asking you.
yes, I understand. But I think you can all get a sense that I have a powerful feeling that there's something much greater than simply science.
Okay,
in the
good
natural world.
presentation. I like that.
Hello. I have to admit I didn't come here tonight with a prepared question, but you really sparked my interest on the whole thing. On your presentation on the origin of us and everything that is in the world, it seems like you based a lot of your presentation on what is already in place, like the elemental atoms and basically a lot of what we're learning in organic chemistry. But one thing that I have not learned in quite a few years of chemistry classes here in a couple other universities is where did the elements come from and how does science explain that?
Very good question. There's a whole process of cosmic evolution. I was talking about one little bit called chemical evolution going from geochemistry to biochemistry. Before chemical evolution, there was a process of what's fancy known as nucleosynthesis, and it occurs in stars. You really should take an astronomy course and learn about stars. They are so incredibly amazing. Stars make the elements. You start with hydrogen, and then the elements fuse together. You make helium, you go to carbon, you go to oxygen, you go to up to higher elements, and then when the stars explode, they make the whole range of chemical elements, and as they're exploding, they then seed the universe with all the elements in the periodic table. The
so
process...
you're suggesting that hydrogen and helium already existed?
Yes. In the way the current model is that the Big Bang, this origin event, and no one knows what came before that, but came into existence, the first elements were hydrogen and helium, that the universe was seeded abundantly with those elements, and then they formed the stars, and the stars emerged from there. It's an amazing story, but there's a lot of evidence for it.
Good evening. Thank you for coming to our little school. I was hoping you could give all of the experts in the crowd and all of us novices a list of maybe your top 5 resources that we could all go and learn more about this topic, be they websites or books or research articles or anything like that.
That might strike some people as a plant, but I won't even mention what I consider the number 1 source in this right now, which is why. But there are wonderful resources. There's the International Society for the Origin of Life, ISOL it's called. They have a wonderful website with lots of resources. There are great books on origin and evolution. There's one by Stanley Miller's student, Jeffrey Bada. Wills and Bada wrote a book called The Spark of Life. There's a wonderful book that I use in my Origin of Life class called Biogenesis by Noam Lahav, published in 1999. And there are great resources on the internet, of course. Many, many research laboratories. NASA has just incredible resources on the internet. And NASA is sponsoring a lot of research in Origin of Life, including our own. So, good question.
You've piqued my curiosity, so I'm going to ask you a question and you can answer this. I am very concerned about, I'm a wordsmith, and I'm concerned about how words that we use every day affect our ability to think. So I'm going to ask you this question. And the people who were around me listening earlier, be quiet. Could you tell us personally what's the largest shadow you've ever experienced? The largest shadow you've ever experienced?
The largest
shadow.
Shadow. Well, I assume it probably would be during a solar eclipse when the shadow of the moon casts over the entire Earth.
Okay. When I ask this question of people to show how their brains are influenced by words, I get answers like that. The largest shadow that I'm aware of that anybody on this planet has ever experienced is called night.
Night, okay. That's a good shadow.
And the reason I bring that up, The reason I bring that up is the simple fact that our ancestors, ever since they were walking on this planet, knew what night was. But at the time they gave it the name, they didn't know the Earth rotated on its axis.
Mm-hmm.
Fortunately, we had science come along and revealed that to us. And I think we're still in the process of being revealed to. Okay?
Sure.
You know, we got a constitutional right. We got gentlemen that wrote the Constitution 250 years ago. Why do we want to assume all the wisdom existed then? Because they didn't know what electricity was. They'd never seen a car, an airplane, a train, a space shuttle, a computer. We're still in the progress of growing. Okay?
I couldn't agree with you more. I think that's a great statement. It's a statement that applies to science. It's a statement that applies to philosophy. It's a statement that applies to theology. And all modes of human understanding, we have to keep growing, and we have to keep the dialogue going. We have to talk with each other and respect each other. We have to think more broadly, think critically, teach our students about science, about intelligent design, about creations, about all the different options. Teach them the facts, help them to come to understanding. This is the only way that our society can progress. Yeah, we are only, if this is scientific progress, we're down here, and this is where we have to go. But isn't it fun to be part of that? I mean, why give? Let's be part of it. It's great fun. Yes.
I would ask you. You mentioned earlier the distinction of consciousness, the development of consciousness in the development of life cycle. As the slime mold crawls across the floor, or the ants harvest their, aphids, what is your distinction for the difference between the slime mold and the conscious development of the human mind? Where's a cutoff of consciousness? Why are we not just responding to chemical signals of light and heat and chemistry and taste and smell? What is the difference between us and the slime?
I think a lot of neuroscientists would agree that we are responding to various chemical and electrical and physiological impulses. However, I think that one can see consciousness as a different level of emergence because it involves self-awareness, it involves analytical behavior. There are certain behaviors that we display in our conversation here, talking back and forth, that ants don't display, that slime mold don't display. And I'm certainly, there's a fascinating idea that comes out of emergence. Let me just present this to you because it's something that may change the way you think about yourself. It's sometimes said that science continually trivializes the role of the human, that we're no longer the center of the heavens. In fact, we're just one small planet circling one small star in one vast galaxy of billions of galaxies, ever more trivial. And then Darwin said we're not even special as animals. We're just one more link in an evolutionary chain. And using that kind of reasoning, it makes us seem sort of diminished. But if you think about emergent complexity, if you think that there is a progress from simplicity to complexity, then the center of the universe is right here. The most complex thing we know is right between your ears. That doesn't mean that's the most complex thing there is, it's just the most complex thing we know right now. And I think that one can argue that in the sequence of complexity, that slime mold, then ants, and then consciousness. It's a higher level. It's the same in character, but it's the higher level. Thank you.
I'm just curious, you mentioned the left hand and the right hand and how there's a left hand preference.
Yes.
What theories are there that explain why, like the left hand over the right hand? Or are there any?
That's a wonderful question. There are two competing theories, as there are in almost everything in origin of life research. There are some physicists that say that the universe is somehow skewed to left-handed amino acids, that there's a cosmic symmetry violation based in what, in technical terms, the CP violation, the weak force. They talk about there being a parity violation that leads to this selection of left-handed amino acids. Most people in organic chemistry don't really accept that. And they say that life could just as easily have been right-handed with amino acids, left-handed with sugars. It's just a chance whichever system of chemicals was the most competitive that outsurvived the others. It's the one that won, and it ate all of its competition. And so if we go to Mars, if we go to another world, and there's life there, it's just as likely to have the opposite handedness. Indeed, some people say that's the most likely way we're going to find life on another world, is to find a world that has the reverse chirality, only possible through a different origin of life.
I'm wondering if you think the scientific search for God is hubris. If maybe God, it's not about faith. If maybe we're supposed to find God at some point, and if searching for God scientifically, empirically, is hubris, or if it is conceivable. Maybe God is not Christian, God is not Jewish. Maybe we need to search for this God so we can find out who he is.
Science can't answer that question of whether there is a God or what the nature of God is. God is, by definition, beyond what we can see in the natural world. Science is so limited. Let's make that clear from the start. Science can only ask very narrow types of questions. We can answer things where you can make experiments, observations, reproducible, verifiable, use the kind of mathematical reasoning and logic. It's this methodological naturalism which people find so daunting in some cases is really just a very limited pursuit. But it's the only way we can ask objective, verifiable questions about the natural world. That's what we limit ourselves to. We don't invoke miracles. We don't invoke God. So I don't think that science can answer that question. I think if you want to know about God, you have to look inside yourself. Now, that doesn't mean that science cannot help inform your faith. If you want to know about the Creator, there's nothing better, I think, than to look at the creation. But as you look, then you have to filter it through your own heart and your own mind and your own soul and come to your own belief through that, through revealed sacred texts, through various other people who are spiritual leaders in your life. Science cannot do that. Science should, and any scientist who says that God exists, or even worse, the scientists who say that God doesn't exist, they're just wrong. They can't say that. It's not a matter of science, it's a matter of faith.
I don't think there's any more questions, so we'll end it there. I don't mean to stop the discourse, though. We have a reception set up with punch and cookies, I believe, so continue talking and discussing. And we have book signing over there, and I'm sure Dr. Hazen would be happy to talk with you more. Thank you.